This is part 2 of our lecture on seedless vascular plant. We are reaching for the sky. In this one, we're going to talk about Moniliophytes, so our ferns and horsetails. The first group of Moniliophytes that we'll look at are the ferns. And I think Moniliophytes now is maybe a term that isn't being used. It arose and I think maybe has passed trendiness, and maybe it's Pteridophytes now for encompassing both ferns and horsetails. We'll see. So if we are drawing our sporophyte here, say we have a rhizome, so I don't forget to draw it, growing underground with all its nodes, producing adventitious roots. And then when it comes above ground, it is going to produce megaphylls, so these large leaves that we know as fern fronds. And those usually will have some kind of compound leaf structure. Oh, what a beautiful fern. I always feel so good at drawing when I'm teaching this class. Just hire a kid to do this for me. So this is a megaphyll, and if we were to zoom in on one of these sporophylls here, one of these sporangia making leaves, so I'm going to look at the underside of it. If you ever looked at the underside of a fern, it has these little, it depends on the fern, but often sword ferns in our area, they have these kind of dusty orange looking dots. So we're going to look at the bottom of that, and we're going to look at the leaf surface, and if we're microscopically kind of looking at a cross section through it, so from the side, you would have this umbrella-like structure, and this isn't always present, but it protects the developing sorus. So a sorus is a cluster of sporangia. And some ferns will have an indusium, which is this umbrella-like structure that protects the developing sporangia, which develop underneath it in a cluster. And you'll see this in lab, it's very fun. Each sporangium is encircled by these large inflated cells. And it's not all the way around, it's a ring that goes around the sporangium. So you have a spherical sporangium that has this ring of larger cells around it. It kind of looks like a caterpillar under the microscope. So that annulus is around the sporangium, and those cells, when the spores are mature inside, will dry out and rip the sporangium open to release the spores. And inside here we'll have it's homosporous, so we have homospores. They're all the same. They'll grow into bisexual gametophytes. I encourage you to look more deeply at what fern sori look like, because they're really cool. Some of them look like this little pile of eggs, and those are all the sporangia and the sorus. Sometimes they'll have an indusium, sometimes not. Sometimes they will line the margin of the fern leaf on the underside, and the fern kind of curls over to protect them. Very cool, distinct structures. Kind of sparks your sense of wonder. Here we see the fern gametophyte. It's a kind of heart-shaped structure. It's just a little flap of tissue. It's photosynthetic. This would have grown from one of those homospores that landed and then grew by mitosis. The cells have multiple chloroplasts inside. You can see them. And then if we were to flip that gametophyte over, you would see these root-like structures, but because we are in a gametophyte, these are still rhizoids. They're not roots. They don't have vascular tissue. None of our gametophytes have vascular tissue. That only happens in our sporophytes. So we have rhizoids, and we also have both antheridia and archegonia forming. I don't think you can see either one in this picture. Here's more rhizoids though. So if we look at our fern fronds, these develop in an interesting way. So they start out curled up in a structure called a fiddlehead. So you can see that here. and they form their leaves starting from the bottom. Those leaves get formed first, and as this fiddlehead unrolls in a process, so this whole process together, the development of the leaves from this sort of base upward is called cercinate vernation. So as it uncurls, it's forming those newer leaves. So those newest leaves are protected as they're developing by this curled frond. So a fiddlehead is a way to protect your kind of sensitive growing leaves instead of just producing them out there because you don't want them to get eaten, right? So you're protecting those newest leaves. So that unfurling of a fiddlehead is called cercinate vernation. If we look at the bottom of a fern, the underside, the abaxial side, you can see the sori. And these sori here are just little clusters of sporangia that are not covered by an indusium. So this is an example of a fern that doesn't have an indusium. Not even, I don't think we can even see it in the younger one. It might have a really thin kind of transparent indusium, but I don't think so. And this is what it would look like if we did a cross section and looked at it microscopically. So we have the leaf, our sporophyll here, and this whole thing is the sorus. It's covered by an indusium, and that will kind of shrink and collapse as these spores mature. Each sporangium is encircled by an annulus, those thickened cells that are full with water that then dry out and shrink, cracking open the sporangium. And inside, all the spores are about the same size. It is homosperous. You can see we have many, many, many sporangia in this sorus. So not only do we have a branching sporophyte, but we have many sporangia in each sorus. Here we can see an indusium covering the sporangia, so it's not present for the whole development, right? That indusium is going to kind of collapse as those sporangia grow up and around it. This is a beautiful picture of what the annulus looks like. So you can see it's just this ring of enlarged cells that once these cells start to dry out, it's going to pull them backward and open up that sporangium, as we can see here. And so that releases those spores. It is a great time to be out right now looking at horsetails. So horsetails have a really interesting feature where they'll have a vegetative shoot that'll be connected underground to a reproductive shoot. And the vegetative shoot has photosynthetic branches in a whorl. I guess I should draw the rest of its stem here. And then it has leaves also arranged in a whorl. But those leaves are pressed to the stem and they're not photosynthetic. They're still megaphylls because they have multiple veins of vascular tissue, but they are not photosynthesizing. Your photosynthetic parts of this vegetative shoot, which I should label, are the branches. Horsetails are pretty fun. If you touch one, you'll see it's kind of like has a lot of friction to it. It's a little bit sharp, and that's because it has silica granules all over it. And so it used to be called a scouring rush. I suppose some people might still call it a scouring rush because of that silica. It's like tiny pieces of glass, so you can use it to scrub pots. So that is attached to the reproductive shoot underground by a rhizome. Here's our rhizome, which would have little adventitious roots coming out of it, roots that emerge from stem tissue. Oh, so beautiful. And then it will also have these whorls of non-photosynthetic leaves. And usually the reproductive shoot is going to be kind of a sickly looking yellow color in sort of the end of mushroom season. And the beginning of kind of spring plant season, people will start posting pictures of these reproductive shoots asking if there's some kind of weird mushroom. So this whole shoot usually doesn't photosynthesize. It gets all of its food from the reproductive shoots. So all of the sugar that gets made in the vegetative shoots. So all the sugar that gets made in the vegetative shoots from photosynthesis gets transported through the rhizome to these reproductive shoots, whose whole function is to reproduce. So this, go away. This is a strobilus. Malfunction. And that strobilus is going to be producing spores. And the spores that it makes are photosynthetic and haploid. And they have these structures on them called elaters. There's 4 of them. And at first they're wrapped around the spore. But as the environment dries out and the humidity is just right, they stretch out away from the spore and kind of dance around to help that spore disperse in the dry air. If it's raining, those spores are going to get knocked down to the ground and will have to germinate close to the parent plant. If we want longer distance dispersal, then these elaters will respond to the right conditions of moisture where it's a little bit drier out and then expand and allow these spores to travel long distances in those air currents instead of ending up close to its parent plant. And this is, so it's a haploid spore, and it's also a homospore because it's going to grow into a bisexual gametophyte. Here we can see the equisetum gametophyte. So equus, you'll see that term for horse, right? So it's our horsetail. The gametophyte is photosynthetic. It's tiny, right? I have never actually seen an equisetum gametophyte. Maybe if I were to dig around in the soil, I could figure out what one looked like. Here we have the reproductive shoot. All of these are, or not, hard to talk today. This is the vegetative shoot, doing the vegetable stuff. Vegetables are food. So our vegetative shoot makes these photosynthetic branches in a whorl. It has non-photosynthetic leaves that are actually still considered megaphylls because they have branching vascular tissue within them, even though they're kind of small and dinky. The reproductive shoot over here, you can see it's drawn in white because it is non-photosynthetic. It gets all of its food through the rhizome from the vegetative shoots. So the vegetative shoots make all the food, distribute it through the rhizome to the reproductive shoots that then make the spores. And here they are in real life pictures. The vegetative shoot on the left is what we usually think of as a horsetail, and then the reproductive shoot on the right, this one actually has more green to it than a lot of them that you'll see. Sometimes they just look this weird sickly yellow. So this one's actually probably doing some photosynthesis, but some won't do any photosynthesis at all. This is a long section through a strobilus. So here we have this T-shaped structure that bears the sporangia. So O4 means it's bearing, right? So we have the sporangia 4 bearing the sporangia. And from the top, this looks almost like one of those rides that has all of the chairs that then spins around at the circus. So they're like chairs on strings, and then it spins around. The sporangia will sort of peek out from the sides of each of these. There's not just 2. It's like a, I don't know, a post with a plate on top that's then holding all of these bags of sporangia. So that's the sporangia 4 holding the sporangia that produce homospores that then are photosynthetic, go out into the world and use their elaters to disperse themselves when the conditions are right. This is a video of elaters in action. Here they are not acting, but I'll link this video in the comments so you can see them move around. One of my favorite parts of the year is when we get to look at these under the microscope. You put them under the microscope and you watch and you put it on a dry slide and you look at them and they have their elaters out and moving around, and then you watch as you put a little drop of water next to them, and as they start to get close to the water, as the water approaches them, they just ball up. And then as soon as that water dries out again, they open. It's really fun.